Good afternoon, everyone. I'm Jane Dixon Weber, the Director of Education and Support Services at the National Fragile X Foundation. I would like to welcome you to today's webinar, Fragile X Genetics and Mosaicism. Today, Brenda Finucane will explain mosaicism plus answer any other burning questions you have from the Fragile X community. I want you to know these webinars are made possible because of your contributions to the National Fragile X Foundation. A little bit of housekeeping. Brenda does have a presentation for us this afternoon. Please use the chat box on the side for your questions or if you have any problems. Now, I would like to introduce today's speaker, Brenda Finucane. Brenda is a licensed genetic counselor and the Associate Director of the Autism and Developmental Medicine Institute of Geisinger Health System in Lewisburg, Pennsylvania. Brenda has a longstanding clinical and research interest in Fragile X and other genetic disorders associated with complex intellectual and neuropsychological symptoms. She serves on the Foundation Board of Directors and on the Scientific and Clinical Advisory Committee of the Foundation. She is also a a consultant genetic counselor for the Foundation. Good afternoon, Brenda. It's great to have you.
Thanks, Jane. Happy to be here, and hello to everyone out there. I have often said that as a genetic counselor, the genetics of Fragile X associated disorders is among the most complex patterns that we see as genetic counselors, and I frequently receive calls and inquiries from other genetic counselors who maybe don't deal with Fragile X as much as I do, just asking questions and sort of going through it. So I want you to know, if your family's out there and you're finding this really hard to comprehend sometimes, and all the various twists and turns that we see in Fragile X, you are not alone. It is really quite a complex inheritance pattern, and it really is something that a lot of professionals as well have to work really hard to get under their belts. So welcome, and today I'm going to focus on mosaicism in Fragile X. Oops, sorry. Okay, before I start, I have a few disclosures. Jane already mentioned that I'm a consultant and I'm on the Board of Directors and the Scientific and Clinical Advisory Committee for the National Fragile X Foundation. I'm also a consultant for LabCorp and also for Ovid Therapeutics, and I work at Geisinger in central Pennsylvania at the Autism and Developmental Medicine Institute, which has a Fragile X Center. So today I'm going to provide an overview of Fragile X inheritance just to start with, to make sure everybody's on the same page. And then I'll talk about mosaicism and what it is. What is the mechanism? How does this thing happen that we call mosaicism? And specifically focus on the different types of Fragile X mosaicism, which involve mosaicism for repeat size and also for methylation. And then I'll talk a bit about what mosaicism means in an individual child in terms of prognosis, and how do we think about that? How do we think about how that child might do when there is evidence for mosaicism? And then we will leave some time for questions and answers at the end, and I know people will be sending them in and have sent them in, and then Jane will read your questions and I'll try my best to answer them. So I'm going to start just briefly without doing a long detailed science lesson here, but just to make sure everybody's on the same page. When we talk about Fragile X, we're obviously talking about a genetic disorder, and we're talking about a condition that involves a gene called FMR1, which is on the X chromosome. And chromosomes are packages of genes. Most people have 23 pairs of chromosomes. And if you look in the body cells, this is what they look like. This is a very old technology called a chromosome spread, where you could actually look at the chromosomes in the cell. This has been around for many decades. And in the cell, they're all sort of scattered around. But when you actually look at them in an orderly way and kind of line them up, you see that they come in pairs. And the reason that chromosomes come in pairs is because we get one copy of every chromosome from our mothers and one from our fathers, and we call them by different numbers. So there's chromosome 1, chromosome 9, chromosome 12, And then, of course, our chromosome of interest are the X chromosomes. And the X chromosomes are special because they're part of a pair that determine whether somebody's a male or a female. In this case, this female has 23 pairs, so 46 chromosomes, and she's got here 2 X chromosomes. They're large chromosomes containing many, many genes. And here's a male who has 1 X chromosome and then 1 smaller chromosome called a Y, which contains genes for maleness and which is primarily found in males. So this is the chromosome of interest, and each of these chromosomes, as I said, are packages that can contain hundreds of genes. So all of these 23 pairs together, there are about 20,000 genes packed into them, half from our mothers, half from our fathers at the time of conception. So when I always meet with individuals who get and families who get diagnosed with Fragile X and they're wondering what this is about, and your tendency is to kind of focus, oh, it's all about Fragile X. Well, no, because that's actually just one gene out of all the 20,000 genes that we have that determine lots of other things about us. So no wonder there's a lot of variability, and no wonder people with Fragile X share a lot of characteristics as well in common with their families, because they have all their other genes that they got from mom and dad. So just briefly, Fragile X genetics, we know it's an X-linked disorder. So the gene that causes Fragile X is on the X chromosome, and we call that X-linked. It's called FMR1, which stands for Fragile X Mental Retardation 1. It was named many, many years ago when that terminology was acceptable, and they're not going to change it, I don't think, anytime soon. So we refer to this as FMR1. FMR1 is a gene that has a section in it which has this repetitive string of chemicals, and we refer to this string of 3 chemicals in a row as a repeat. So the chemicals cytosine, guanine, guanine are kind of in a triplet, as we say. The 3 of those go together and they're repeated over and over again, and we call that a CGG repeat. And in this FMR gene, there is one section that has a whole repeated section of CGGs. And in the average world, most people have an average number of repeats of about 29. And when you get a lab report back and you've had a Fragile X analysis done, because women have and females have 2 X chromosomes, you'll get 2 numbers for a female, whereas if you get a result back on a male, you're only going to have 1 number. So what do these numbers mean? Well, any result that's under 45 means it's negative for Fragile X associated disorders. It's not an expanded gene. It's the gene the way it usually is, and it hasn't expanded into a premutation or full mutation. There's another category that I won't go into a great deal about, but there are a number of people, in fact, it's about 3% of the population, that have somewhat expanded FMR1 genes, and we call these intermediates, and they have CGG repeat numbers from 45 to 54. And these typically do not cause any kind of problems. They don't cause Fragile X syndrome. They don't expand up to a full mutation. They don't cause FAXTAS or FAXPOI, those associated disorders, but they're somewhat expanded. And so when a genetic counselor meets with somebody with an intermediate, we tell them to keep an eye on it, because over multiple generations, that could expand little by little into a premutation and then ultimately may become more unstable and may lead in later generations to Fragile X disorders. So today I'm going to focus on mosaicism in premutations and full mutations. And premutations, by definition, are expansions of that FMR1 gene where there are anywhere between 55 but under 200 CGG repeats, and full mutations have over or equal to 200 repeats. So again, if you have under 45 repeats, a person is not at risk for a Fragile X associated disorder. If you have an intermediate, there's really no increased risk for that per se. The gene might be very stable, and that might just be a family trait, but we like to keep an eye on it from generation to generation to generation, because little by little, it might become a premutation down the line. Premutations have a range between 55 and less than 200, and there are a lot of reproductive considerations there, because premutations can expand to full mutations in the next generation, and also because of the Fragile X-associated disorders, FAXTAS, or the Fragile X associated tremor ataxia syndrome, and the Fragile X primary ovarian insufficiency, which are two conditions that are found in people with pre-mutations. And then finally, full mutations is when the number is 200 or more, and we see Fragile X syndrome and developmental concerns. So it's important to set the stage around methylation, because when I talk about mosaicism today, there are different kinds of mosaics that we can see, and one has to do with the size of the gene or the expansion. So, is it a full mutation? Is it a pre-mutation? But there's another type of mosaicism that has to do with methylation. So let me walk you a little bit through that. So FMR1, as I mentioned, is a gene that it's found in everybody in the population. It's just that in Fragile X families, it's expanded bigger than is found in the general population. And normally the function of this gene is to produce Fragile X MR protein, so FMRP. And FMRP is a protein that is expressed in the brain and other body tissues before birth and then throughout life. This is really a very important protein for brain functioning. And so if you don't have FMRP, your brain is not going to function the way one would expect. And when you have FMR1 full mutation, so when you have over 200 repeats or more, the gene undergoes a chemical change when it gets that large that we call methylation. And methylation is just a chemical change that happens that silences the production of FMRP. So when you have a very large full mutation and it goes to 200 repeats, it gets so large that this chemical change happens, and we call that methylation. So Fragile X syndrome is caused by lack of FMRP, and that's essential for brain functioning, and it happens because the gene has expanded or is so large that it becomes silenced or methylated. And it's important to note that the Fragile X symptoms and the severity of Fragile X is really not correlated with the size of the full mutation. So you can have a full mutation in a child who has 250 repeats, and he or she is not producing any FMRP on that from that gene. You could have another person who has 1,000 repeats, so a much larger full mutation, but it doesn't matter. They're still not producing FMRP. So the net effect is the same. So I often get asked that question, does it matter whether the size of the full mutation is 200 or 1,000 or whatever it is? And it doesn't. Once it's over 2,000 and methylated, it's not producing protein. So the effect is the same, whether it's bigger or smaller. So it's important to note that Fragile X pre-mutations, which are associated with those Fragile X-associated disorders, do not become methylated. So when we talk about methylation, we're only talking about full mutations. And it only happens because in full mutations, they're so large, they become silenced and methylated. So pre-mutations, if they're under 200 repeats, by definition, they don't become methylated, and so therefore they do produce FMRP, and that's why pre-mutations do not cause Fragile X syndrome. Only the full mutation will cause Fragile X syndrome. We do know that, though, that these pre-mutations are larger than the usual FMR1 gene, and because of that, they get kind of clumsy when you go to try to translate these large pre-mutations into a protein. And so what happens is there's kind of an intermediate chemical that happens when you go from the gene to producing its protein, and that chemical is called messenger RNA. And when you have a pre-mutation, because it's larger than it should be, it causes more messenger RNA to get produced, because the gene is really attempting to produce FMRP, and it's just not as efficient at it because it's a very big gene. What happens then is messenger RNA is toxic to cells, and we believe that is one of the primary reasons or causes for the associated disorders of FAXTAS, Fragile X tremor ataxia syndrome, and FAX-POI, which is Fragile X associated primary ovarian insufficiency. So again, it's like having two different diseases in one, where Fragile X syndrome is caused by the lack of FMRP, but these other pre-mutation disorders are caused by messenger RNA toxicity. And so here's a graphic. I thank Dr. Rodney Hagerman from many, many years ago who shared this graphic with me, and I've sort of edited it somewhat to simplify. But what we have here is, you know, on the left side, you'll see the non-Fragile X-associated disorders range, and you'll see that there's the gene FMR1 on the left and the usual number of repeats, less than 55. It's producing what we would expect, the expected amount of messenger RNA, and then that does produce Fragile X protein, or FMRP, and the outcome is that you have somebody who's not affected by a Fragile X associated disorder. In the middle, you have people with pre-mutations, so they have an expanded gene. It's larger than usual, and as I mentioned, because the gene is a little unwieldy, it's got a little difficulty producing that protein, but it does end up producing it. But in the meantime, it's producing lots of messenger RNA in order to get the job done, and it's that extra messenger RNA that seems to be causing FAXPOI and FAXTAS and some of the other symptoms potentially. And they certainly do produce protein, but probably not at the same levels that you would see in somebody who doesn't have a Fragile X expansion. And then finally, on the right side, you have a full mutation with repeat sizes of 200 or over. And when it gets that large, you're really not producing adequate messenger RNA at all, and it's not producing protein, and so therefore you have Fragile X syndrome. So with all of that complexity as background, which may be review for some of you, but you know, there's a lot of moving parts here when we talk about Fragile X. There's the differences between males and females. There's methylation and not methylation. There's pre-mutations and full mutations. So there's a lot to take in. And now, to add on top of that, a significant number of males and females who have FMR1 expansions, whether it's a pre-mutation or a full mutation, have what we call mosaicism. And there have been a number of studies on this over the years, and they can range from 6% to 41%. So there's quite variability in the studies that have been done. But the Am I still on?
We lost you for a second, but I can hear you again.
Yeah, I can hear you now.
Okay. So mosaicism, to start, is by definition, mosaicism means that there are two or more different patterns of genetic information in the same person. And mosaicism is not just something that happens in Fragile X, as I'll show you. It happens naturally in nature. It happens in other genetic disorders, etc. So I'll talk about that shortly. But again, a significant number of males and females who have FMR1 expansions have mosaicism, where they don't just have one repeat size, they may have multiple repeat sizes, and they maybe don't have just a whole population throughout their bodies of methylated full mutations, but they may have some that actually are not methylated. So there's some mosaicism going on in there. So the mosaicism in Fragile X, we're primarily talking about mosaicism for repeat size, and that's by far the most common type of mosaicism that we see. And then also mosaicism for methylation, and again, I'll explain those shortly. So when we think about mosaicism, the word obviously comes from mosaic, and a mosaic is a picture or a pattern produced by arranging different small pieces of material like stone, tile, or glass to make a bigger picture. And the reason we think about mosaicism when we're talking about this concept in genetics is because our bodies are made up of trillions of cells, and those trillions of cells each of them have their own packet of genetic information in those chromosomes that I showed you. So 20,000 genes. So every single one of those trillions of cells has a packet of genetic information, and it should be and is typically the same from one cell to the next. So it's the same packet, it's just been reproduced throughout the body. However, we do know in biology that mosaicism can also occur in body cells, where some people actually have different information in some cells than they have in others. So they have two or more different types of genetic information in their cells, rather than having just the same copy of the same genetic information repeated over and over again. Mosaicism can be seen, as I say, in nature, in animals, and we can look at these animals here and immediately kind of see differences with them, where obviously some of their cells are producing different colors and striking different colors than the rest. So we see the bird here, the parakeet, that's got two different colors along the midline there. Same thing with the puppy and the cat. In fact, calico cats, which have multiple beautiful markings, it's really because there's different parts of their body cells that are producing, that have genes activated for different types of coloring, and so they are mosaics. And so when we see these types of patterns, we see mosaicism. People can have mosaicism too, and sometimes you'll see mosaicism manifest itself as different color eyes, either two different color eyes or differences within one eye. This little girl has a mosaic form of Down syndrome. So Down syndrome is a condition, as many of you are aware, that can cause a wide range of intellectual and physical differences. But a small minority of people who have Down syndrome don't have an extra 21 chromosome in every single body cell. Most of them do, but a small percentage have some body cells that don't have the extra chromosome. So they would have the typical number of 23 chromosomes, but then they'd have others that had 47 chromosomes, the extra 21 indicative of Down syndrome. And so this little girl has mosaic Down syndrome, and so there's many other genetic disorders as well that have mosaicism in addition to Fragile X. But of course, because we can't get off the hook easily, Fragile X mosaicism is far more complicated than most other types of genetic mosaicism. So one other quick fact, human females, whether you're a carrier or not a carrier, all females in the human race are naturally mosaics. We are all mosaics for genes on the X chromosome. And that's because in every one of our body cells, we have two X chromosomes, and one of them is randomly inactivated. And the reason they get inactivated, it's nature's way of ensuring that only one X is going to be active in any given cell. Males only have the one X. So if females had both X's active, then, as I say, we would be entirely too fabulous. So nature has figured out a way in every cell to inactivate one of those X chromosomes, but it's random. And so in the trillions of cells in females, half of them have one X inactivated and the other half of them have the other X inactivated. So females are all mosaic. We have at least two different cell types in our bodies. So let me walk you through how mosaicism happens. And again, this is where it starts to get a little complicated in Fragile X. But just generally, what happens with mosaicism is that if you think about conception and a sperm and an egg getting together and forming that first cell that is going to go on and form trillions of cells to make up a new baby, at conception, that happens from one cell dividing into two, and then those cells dividing into two, and so on and so on. It's pretty miraculous when you think about it, that a human being can start from one cell and then through all those dividing into twos end up with trillions of body cells. So there are trillions of cells in the human body, and they get there by division. What happens if you have a premutation? Well, a baby who is conceived with a premutation, who would have gotten that, let's say, from, if it's a girl, could have gotten that premutation from mom or dad. If it's a boy, would have gotten the premutation on the X only from mom. That first cell has a premutation. What we see is that first cell splits into two and then splits into two again. And what you have is a baby typically that has every body cell with the same size premutation on one of the X's. If it's a girl, there are two X's, so she'll have another X that in almost all cases does not have a Fragile X expansion. So we would say that this is pretty typical, that usually when people find out that they are carriers of an FMR1 premutation, when they get their lab result back, it has a number on it, one number for males, two for females, and it tells you that, oh, one of these X's, or the sole X in a male, has an expansion in the premutation range. What can happen, though, is you also might have a full mutation. So that first egg and sperm coming together, you may have a child who's conceived with a full mutation. But again, the full mutation will divide again and again, and then you end up with a baby. In most cases, with full mutations, there's not mosaicism, and it's just that every single cell in the body has the same full mutation and it's the same size. So non-mosaic means, every one of those full mutations looks the same as the other ones. What happens in Fragile X is you may start with a premutation, and when it starts to divide, you can have an expansion. This is after a child's conceived, so it's not at the time a child's being conceived, which is often when the expansion happens. It can also happen after a child's conceived. So a child's conceived with a premutation, and then there's a split somewhere early in development where a larger premutation occurs because it's expanded somewhat. And then all the cells that go on to divide from that larger premutation will have the larger premutation. And all the ones that stayed stable from the original premutation will have that size. So you'll have a mix of cells. And what can happen is you may have a couple of more of these events where the larger premutation expands a little bit further. So you may end up at the end of the day with a child who's got certain body cells that have different numbers of FMR1 repeats, and this is what we call mosaicism. And in this case, it's mosaicism for different premutation sizes. So in this example, none of those expansions went to a full mutation. It's just all different premutation sizes. And so what you might find if you get a lab report that gives you multiple different sizes of premutations, well, we say that's mosaicism for premutations. The same thing can happen with a full mutation. And for this example, I show that the first division seems fine. There's no expansion. But it might happen in a later division. So the first division is fine, but now in the second division or further on down the line, there's an expansion from the full mutation to an even larger full mutation. And so again, what you'll have is a baby who's born who's got different populations of cells in the body with different sizes of full mutations. So the plot thickens even more when you have a child, for example, who is conceived who has a premutation, and some of the cells, when they go on to divide, just remain as a premutation, but then you may have a jump or an expansion to a full mutation. And again, what happens as those different cells go on their merry way and divide is that they will have the size that they were originated with. So you can have a whole population and a mix of cells where there are some that have premutations and some that have full mutations. And in that case, it's mosaicism for a mix of premutation and full mutation sizes. So you're getting the drift here, I imagine, that, when you have different sizes, this is what we're talking about in mosaicism, that it's not just the one length, the number of CGG repeats in every cell of the body. Because of the way that the FMR1 gene can expand, you can see different versions, if you will, or different types of premutations and full mutations. And sometimes you can have just premutation mosaicism, sometimes just full mutation mosaicism, and then sometimes this mix of premutation and full mutation sizes. So when we're talking about mosaicism like these examples that I just gave you, we're talking about repeat size mosaicism. So it's the size of the expansion. And so we call this a size mosaic, and it's got to do with just different sizes of the FMR1 expansion and how many repeats you have in different cell lines that have grown from that first cell. Now, because again, Fragile X is very complicated. It's not enough to just say we have size mosaicism. We also have to have this other wrinkle that we call methylation mosaicism. So remember I mentioned that full mutations, when they get big, and so by definition 200 repeats or more, they're so big they become methylated or silenced. So it's a chemical change that happens. And that only happens in the full mutations, not really at the premutation level. So we're not talking about methylation when we talk about premutation. But typically, if you've got a child who's conceived with a full mutation, it's usually methylated, meaning that it's a large mutation and now it's been silenced, it's not producing FMRP. And usually what happens is it gets passed on, it doesn't change, and so eventually all the cells in the body have a methylated full mutation, and that's typically what we see in people who have Fragile X syndrome, is that they have a methylated full mutation on one of their X chromosomes. But again, there's always a wrinkle. For reasons we don't really understand very well, there are very occasionally cases where we have a full mutation. It's a very large 200 or more repeat expansion, but it's producing some protein. It didn't get completely methylated, so it didn't really get totally silenced. And so we call that an unmethylated full mutation. So this is not at all as common as those variations in size, but we do know that it can happen. And what happens is that you may have a child conceived with a methylated full mutation. It's going on its way of dividing, and then somewhere along the line, there is an unmethylated full mutation that evolves out of this process. And then you might have a child who has a mix of cells that are methylated and unmethylated. So they have a Fragile X full mutation. There's no pre-mutation involved here, but a proportion of those cells are actually able to produce protein because they are unmethylated. They haven't been silenced. And so we call that methylation mosaicism. So that's a lot of different variables going on, not to mention that we've got females and males and different effects that go on according to gender. So when we think about this, we can say, well, if you've got mosaicism for different pre-mutation sizes, there's no full mutation involved. Let's say you have a woman and she finds out that she has a pre-mutation of 65 and then she has a normal X chromosome with a CGG length of 29, and then they also find another pre-mutation length, but it's 120, let's just say. So she's got mosaicism all within the pre-mutation range for the pre-mutation. She's got a 65 and she's got 120. Well, in that case, the mosaicism, the main thing that impacts is some reproductive considerations. Because if a woman didn't have mosaicism and just had a pre-mutation of 65 repeats, for example, the chance that that would expand in her child or the next generation to a full mutation is relatively low. We're talking about under 5% chance. But if she's got 120 repeat, then it's virtually 100% chance that that particular X chromosome is going to expand into a full mutation of the FMR1 gene. So as a genetic counselor, and if I'm meeting with a young woman and she says to me, I have mosaicism for different pre-mutation ranges, what I'll say to her is, what's your top range? Let's look and see what the highest one is. And then that's where I'll calculate the chance of expansion just based on the highest one, because we certainly don't have any way of figuring out which egg is going to have what level of expansion. So the different pre-mutation sizes really don't impact prognosis in a way. They just affect the chance in your next generation that you're going to pass on a full mutation or an expansion. The other type of mosaicism I talked about was if you have different full mutation sizes. So let's say, and this happens actually not infrequently, that you may get a result where they see a whole range of full mutation sizes, let's say in a boy with Fragile X syndrome, and they're all full mutations, anywhere ranging from 250 to 1,000, and it comes out a bunch of different fragments way up in that high range of full mutation. There are no pre-mutations here, it's just a bunch of different full mutation sizes. Well, as I mentioned before, full mutation is a full mutation. It doesn't really matter about the size. Once it's over that 200 level and it's methylated, you are not going to produce any protein. So it doesn't matter that they're different sizes. And the mosaicism in that case would have the same range of outcomes as any male or female with a non-mosaic full mutation, because it doesn't really matter. If it's all full mutation and they're all different sizes, it's just like having a plain old non-mosaic full mutation, because at the end of the day, there's no protein being produced. Where it gets a little more, I guess, ambiguous is when you have a mix of premutations and full mutations in the same person, or they might have methylation mosaicism. So they have some cells that have full mutations that are fully methylated, but others that are producing a little protein. And there are a couple of things to think about there. If you're talking about a child and you're wondering, well, is this person going to be less severely affected than usual in Fragile X because we know he or she has premutations? Well, there are a few things to consider. We do know that if there are premutations within the mosaicism, and they're mixed in with full mutations in different cells, there is the potential for some protein production or FMRP production, and we could say theoretically there's possibly a milder effect that we could see in a male. However, there's a wide range, and for any individual child, just because he or she has mosaicism, we really can't say, oh, it's going to be milder, because it's an entire range. Also, when it comes to females, I mentioned that all females, regardless of Fragile X status, naturally, we all have X chromosome mosaicism. So when you say that you've got mosaicism for Fragile X premutation, full mutations in a female, the prognosis really isn't going to change that much, because you still already have one extra X chromosome that's producing protein anyway in a female, and so it really doesn't change things that much. We're all mosaic to begin with. Females already have a second X chromosome that's randomly producing protein in every other cell, 50% of the cells. So having a little bit of a premutation cell line in there with the full really is not going to appreciably change the range that we see. It could be anywhere from very few effects to very significantly affected. So we think about mosaicism and prognosis, and I do think that there are a fair number, not just in Fragile X, but a fair number of misconceptions about this. And I'll give you an example from Down syndrome. Much more is known about Down syndrome mosaicism than Fragile X mosaicism. Down syndrome has been studied for many, many more decades than Fragile X. There are a lot more affected individuals. The mosaicism is much more straightforward than in Fragile X. And we know that mosaicism occurs in many different genetic causes of developmental disabilities and other disorders. So in Down syndrome, what's been shown is that if you took 100 people who have Down syndrome who have mosaicism, meaning that some of their cells do not have the extra number 21 chromosome, and you compare their functioning to 100 individuals who have full Down syndrome, where every single cell in the body has an extra 21 chromosome, there is a group difference. The group that has mosaicism in Down syndrome is somewhat higher functioning. There's still a range, but their mean or average IQ is somewhat higher than the group that doesn't have mosaicism. And we also know that sometimes the physical manifestations of Down syndrome are somewhat milder and subtler in the group, as a group, those who have mosaicism versus those who do not. So as a group, if we look at large numbers of people, you can see those differences between the groups. However, when you start to think about an individual and say, oh, is this baby now with mosaicism going to be high functioning? We cannot answer that question. And here are a couple of reasons why. Because the degree of mosaicism that we detect is being detected in the blood. So it's a blood sample. Blood does not necessarily reflect the same level of mosaicism in the brain or other body tissues. I mean, just because we have a blood sample that says there's a high degree of mosaicism and there's a lot of premutations here, that doesn't mean that in the cells that went on to the brain, there couldn't be mostly full mutations, which could mean that the child's going to have very much a similar outcome to anybody with non-mosaic Fragile X. And so, for example, and I use these kind of very simplistic pictures, different body cells have different patterns of mosaicism. And the only one that we measure is the blood, but what we're really interested in Fragile X and Down syndrome and other conditions is the brain, like what proportion of brain cells actually have FMRP expressed. And that can really be quite different than the blood, and we have no way to predict what's going on in the brain, frankly. And you could have a child who's really got a very high ratio of premutations to full mutations in a blood sample, but is very much affected like anybody else with Fragile X because the brain has primarily full mutations. It doesn't have quite as many premutations or maybe no premutations in the brain cell. So that's why, as a group, you're always going to find people who, kind of lucked out in a way, and their brain cells did get the premutation and got some FMRP production, but you'll also find those who really didn't, and even though they have mosaicism, their brain really didn't benefit from that FMRP because it's a different tissue. And you put all of those people together, and then as a group, you may find that, oh yeah, as a group, their average IQ is somewhat higher than the individuals who don't have mosaicism, but again, you can't use this for a prognosis in any different child. And in fact, for any child with Fragile X full mutations, even without mosaicism, there's such a wide range. I know many people, and I'm sure others do as well, where the child may have a methylated full mutation, it's not mosaic, the same amount of CG repeats in every single one of those cells, and they're quite high functioning, and it's not explained by mosaicism. And then you might meet somebody who's got mosaicism and they have a good mix of pre-mutations in with full mutations, so theoretically they're producing some protein, but they may be very severely affected, and even more affected than somebody who has a full mutation Fragile X without mosaicism. So in conclusion, we know that mosaicism for different repeat sizes and or full mutation methylation patterns, it's pretty common in Fragile X, almost half. I would say probably closer to a third if you put all the different studies together. We know that females are naturally mosaic anyway for our X chromosomes. So really, there's already a very broad range of functioning due to FMRP because we produce FMRP on the non-Fragile X chromosome. So therefore, having some mosaicism for the Fragile X chromosome really doesn't tremendously contribute to a difference in outcome as far as we know. And again, there are very few studies on females related to mosaicism. So when we talk about prognosis, we're primarily wondering about boys, and we know that when mosaicism in boys involves a mix of premutations and full mutation sizes and or differences in methylation patterns in a full mutation, there's the possibility that they're producing some protein. And when you have the possibility of producing some protein, there's always a chance that this person could develop and have much more mild effects of Fragile X than a boy who doesn't have mosaicism. And as a group, we do know that boys with mosaicism may have milder Fragile X effects, but at an individual level, we just unfortunately can't predict that, and your child will be your guide. I think this is an important thing to know. I talk to parents of, young children, very young children who have mosaicism, and, just like any parent, they're thinking, they're hoping for the best for their child, trying to maximize development. But you should always keep an eye on how your child really is developing and not assume that there won't be any problems because of methylation. Certainly, there's no guarantee that there is protein being expressed in the brain. And likewise, for parents who have children who do not have mosaicism, that does not mean that those children, that's the majority of kids with Fragile X who don't have mosaicism. It doesn't mean that they would be on the more severe end because they don't have mosaicism. It just means that there's they just have the same Fragile X CGG repeat number in every single one of their cells instead of that type of variation that we see in mosaicism. So that's what I have for you today. I want to acknowledge my colleagues out here in central Pennsylvania who help to run our Fragile X clinic and center. And Jane, I know you have some questions probably coming in, and I'll be happy to answer them as best I can. And I want to, again, thank the National Fragile X Foundation and for all of your donations that bring these webinar series to our Fragile X community. I think it's important for people to have information, and the fact that we can have webinars and then archive them is really great. And so if you haven't checked out the other webinars in the archive, there's a lot of rich information there on every subject under the sun.
Well, great, Brenda. Thank you. That's, I learned something every time. One question that just popped up when you were talking about the difference, I know that I've seen some people post their test results on Facebook, and they'll have 3 numbers. And so it looks like, they'll say, oh, that's mosaic. But so on the test results, do they always put whether or not it's methylated?
That's a great question. They do not always put whether it's methylated. So there are 2 different tests. Traditionally, there are different tests now that can sometimes be used, but let's just stick with sort of the standard. There's a test called PCR, which is polymerase chain reaction, and the other one is a southern blot. And they usually go hand in hand, and one will give you the number of repeats, and the other will test for the methylation. And so, things have changed, and labs differ in how they report these out. So a number of labs will do those 2 tests, and they'll start with the one, and if it turns out that the number of repeats is in the full mutation range, it'll reflex to checking on that methylation status, or vice versa. And so you'll get the 2 numbers. It'll say, this person has, 275 repeats and it's fully methylated. You have other labs will just report that it's methylated, and they may not give you the number of repeats even, especially if in some people with Fragile X, there's a large smear of repeats. In other words, there's lots of different fragments. They're all in the full mutation range. You're not going to pull out one particular number there, but they know it's like in the full mutation range and methylated. So I think the take home is that lab results are going to vary by the lab, and some of them do not report your number, but if you read the very long 3-page description that's in there, they often will say this is a methylated full mutation, or they may report the number, but they don't necessarily report the methylation, but that doesn't mean they didn't, assume that it is methylated. So I would go over those reports with a genetic counselor. With regard to having 3 different numbers on there, so that we see to me, anything more than 2 in a female and more than 1 number in a male would mean we're talking about mosaicism. So if it's a female and there's 3 different numbers reported, my hunch would be the first number would be the non-Fragile X, so it's usually a 29 or a 30 almost all the time, or something under 55. And then the second 2 numbers would be different, either pre-mutation sizes or full mutation sizes, or a pre-mutation full mutation mix. And so that's something that would indicate mosaicism in a female. You could also have a male who's got 3 numbers, and it just means that he's got 1 X chromosome, but it's got 3 different sizes of mutations in there, full mutations or pre-mutations or a mix. And so those numbers are important, and it's important to go over those lab tests, again, with a qualified genetic counselor or a physician who knows about genetics to kind of understand what those numbers mean.
Great. Thank you. Here's another question for you. So my son is full mutation / mosaic, as well as he has autism. At 13 weeks pregnant, I had a CVS test, and the test came back negative for Fragile X. How does this happen?
So, well, I don't, is he third, do we know how old that person is now?
Can you type in how old he is now?
And the reason I ask that is, in the early days...
He's 7 years old.
Oh, he's only 7 years old. Okay, so then I was going to say many years ago when CVS was just starting for Fragile X, there were issues in confirming methylation, but that shouldn't be happening recently. So I don't know how to answer that. Did they do the specific test for Fragile X is my first question as a genetic counselor. There are many different types of tests that they do during amniocentesis or CVS, but they do not routinely do Fragile X prenatal testing unless it's specifically indicated. A CVS that a woman would have because she's 38 years old, for example, is not going to check for Fragile X. You would have to have a confirmed family history. So, you know, I don't know the specifics of the case, but again, my first hunch would be that they didn't check for Fragile X if they didn't know they were looking, and if they specifically were looking for Fragile X, the other explanation could have to do with the mosaicism, that when they were doing the CVS, the cells that they look at for CVS, or chorionic villus sampling as it's called, really check into the cells that make up the placenta. And sometimes those cells may not have an expansion. And it's possible, if he's mosaic, that the cells that were in the sample that they took from the placenta were not cells that had a Fragile X full mutation, but had a you know, something that was in his mosaicism. So without knowing specifically, it's hard to say. But again, I'd be happy to follow up offline if you want to email the foundation. I can get back to you if I know the specifics.
Yes, if you'd want to email me some more information, I'd be happy to follow up with that. Okay. Females with a mosaic pattern, do they exhibit higher anxiety? There's a typo. Social. Do they exhibit higher anxiety / social anxiety? And what therapy is most effective, especially for females?
Well, the first thing to say about females is, you know, we do know that anxiety, and particularly social anxiety, is quite common among females with Fragile X syndrome. There certainly has not been research to be able to confirm that there's any higher rate of this going on in females with mosaicism versus those without mosaicism. We see it certainly across the board in many females who have Fragile X syndrome. And then, as I explained before, all females are mosaic in a way for Fragile X because we have that second X that's producing protein. So there shouldn't be really a substantial difference going on in the way these girls present, but there's such a broad range in females who have a full mutation, whether or not they have mosaicism. So my personal thought would be, well, first of all, there's no research that has been done on that at all. And the second thought is, you know, all females are mosaic for Fragile X because of the nature of the X chromosome. And so that what you're probably seeing is more just something along the continuum of what we see in females with Fragile X generally. And I wouldn't necessarily want to say it's really because of the mosaicism. If anything, it shouldn't be related to that at all. With regard to any interventions for anxiety, again, I think that's a webinar in itself. Anxiety is a well-known aspect, and social anxiety, of Fragile X. And I'm not sure what webinars that we have on that topic specifically, but there certainly have been a number of articles written on the topic. So, you know, what I would say is to try to find some of the resources that are out there on social anxiety. A number of people have written about this in the Fragile X community, and there should be some strategies there. Certainly it amounts to, you know, some behavioral interventions and special education and, you know, medications at times. And so these are topics that the National Fragile X Foundation has compiled a number of articles on over the years. And sorry to throw everything back on you, Jane, but perhaps that person asking the question, you could direct him or her to the some of the resources that I know are out there from my colleagues, Marcia Braden and Karen Riley and Tracy Stackhouse and Sarah Scharfenegger and others.
Yeah, shoot me an email. I do have some articles I can send you. My son was tested in 2012. Would the lab results clearly state if he was mosaic?
If mosaicism is detected, and again, there are many different labs out there, so, and the quality of reports from different labs is almost as variable as the clinical features in Fragile X. But if a lab, a reputable lab, identifies mosaicism, they typically would report that. That's a reportable finding in Fragile X. So I suspect that they would have said it. If it does not say mosaicism, my assumption would be that no mosaicism was detected. Now, that being said, as I said before, these tests are being done on blood samples. So, it is only going to be able to detect mosaicism in blood. And so everybody who gets a report that doesn't mention mosaicism, which is the majority of people with Fragile X syndrome, then, we're making an assumption that they're not mosaic, and they probably aren't, but of course we're not checking any other tissues. So I would assume if it doesn't say mosaicism, then they didn't detect any.
Okay. This one I'm not for sure about. I might need some more information. If the genetic test indicates repeats in the FACS-TAS range, is there a way to predict whether FACS-TAS will present later in life? So if I'm reading this correct, I would say that if the genetic test indicates repeats that are less than 200.
Yes, and in the pre-mutation range. So yes, so the pre-mutations are associated and causative of FACS-TAS. And again, there's no hard and fast correlation between the numbers in the pre-mutation range and age of onset of FACS-TAS. So, we do know that males, at least half of males who have a Fragile X expansion in the pre-mutation range will eventually develop FACS-TAS, but if you look at those males, there's an average age of onset around 60, but it can be widely variable. It can be much later, or it can be somewhat earlier. There's nothing about the number that you get in that pre-mutation range that can help predict when that age of onset will be.
Right. They haven't been able to identify any correlation like that.
Not in that, not with age of onset, yeah. And, the thing to keep in mind is there have been decades of research on Fragile X syndrome. We've learned a lot. There's still a lot more to learn. But FACS-TAS and FACS-POI, the related or associated disorders, really we're in our infancy in many ways in learning about them. There's a smaller body of researchers looking at it. They were discovered much more recently. And so we still have a lot more work to do there. And hopefully someday we will be able to look at that. But, I mentioned before that when we think about Fragile X, it's just one gene out of all of our 20,000 genes. And some of that variability, and likely the majority of that variability in age of onset for things like FACS-TAS comes from our background, our genetic background genes. So why does somebody have an age of onset of 80 and somebody else has an age of onset of 55 for FACS-TAS? Well, they both have the same pre-mutation, same size pre-mutation, let's say, but it could be that there are other genetic factors in their background that are determining how soon they get the symptoms of FACS-TAS. So our other genes are definitely playing a role in modifying how these various Fragile X symptoms come out.
Great, thank you. All right, here's another take. Is it common for mosaic Fragile X children to be nonverbal? My son's numbers are in the pre-mutation range, 130 to 230, and full mutation 170 to over 1200. His DNA was partially methylated.
Mm-hmm. And do we know how old he is?
Can you tell me how old he is? Let me see if she types it in. He's 8 years old.
8 years old, and he's nonverbal still at this point. So, we do know just across the board in Fragile X in boys that language skills are quite delayed. 8, is more delayed for language. You should have, we typically see a few words by then at least. But again, I think this kind of illustrates exactly what I was saying about mosaicism. You can get a blood test that says that there is some protein being expressed, that there's cell lines with pre-mutations showing in addition to the full mutations, but we don't really know what's going on in the brain. And so within the whole group of kids who have mosaicism for full mutations and pre-mutations, there are going to be some, yes, who really their brains are benefiting from that protein. They happen to have it expressed in their brains. And then you'll have others who really are more typical of kids who don't have mosaicism. So he's still 8. There's still certainly room for improvement and room for growth there. So you won't know, ultimately where he'll be for quite a while. But again, I think it kind of shows that you can't predict just based on the mosaicism that he was going to be an early talker, for example, compared to other boys with Fragile X syndrome, and in some cases may be nonverbal or a later talker than boys who don't have mosaicism.
Okay. We're at 4 o'clock. Do you have time to answer a few more questions?
Sure.
We can maybe 5 more minutes, a couple more questions. My son's test says 400 to 1,000 repeats, normal and abnormal methylation. PCR is 145 and over 200 repeats. And my other sons read similarly, but with different numbers. Is this a repeat size mosaic or a methylation repeat? Probably a methylation.
Or yes, it sounds like a methylation mosaic, but that PCR report of 145 to over 200, is that what you're saying?
Yes.
Yeah, so I'd have to see the report actually, and again, I'm happy to sort of look at that, but what it could be, I'm not seeing the report, but it could be that there is a pre-mutation, full mutation mix there, and then you also have methylation mosaicism that's being detected as well. So then, and that can happen. So it certainly sounds like it's mosaicism. It sounds like they're identifying that there is some lack of methylation going on, which means there may be some protein production.
Okay. So if you want to send me your test results, we can take that further. So here's kind of let me just see if this is kind of a variation on what I mentioned earlier. My son has 3 scores, 2 pre-mutation, 1 full mutation. He's 10 and starting to read. Is that a good sign that his learning will continue, or will he plateau, or is this unknown? Just trying to see, what you think about his progress.
Yeah, so that, again, that's great to hear that he's starting to read at 10. I think that the answer C is the right answer, that it's unknown, and really he's going to be your guide more than his lab test is going to be your guide, because as you can see, I could have gotten that same sort of report. I have a child with Fragile X who's starting to read at 10, and it could easily have been from the parent of a boy who doesn't have mosaicism. And again, that happens also without mosaicism. So of course it's a good sign that he is starting to read and looking at that. But I think that's where the developmental specialists are going to be your best allies in sort of seeing what are the strengths and weaknesses that your son has in his learning, and how to work to really kind of compensate for some of the things that he struggles with and really maximize the things that he's very good at. And it's really not going to be determined by his lab test. When I think about mosaicism, there's 2 ways you can come at it. I often hear from parents of new babies, and they are very young children who have developmental delay, and then they find out that the child has mosaicism, and then of course, there's a range even without mosaicism, so nobody's got a crystal ball, and I try to say, well, we need caution here. Just because there's mosaicism does not mean that this child's going to speed past everybody else without mosaicism. The other angle that I often see is I might run into a teenager or an adult who's quite functional with Fragile X, good verbal skills, has some good work skills, maybe some social anxiety, and really is doing quite a bit better than typical of boys or with Fragile X syndrome, or young men with Fragile X syndrome. And then I find out, oh, he's got mosaicism. And so then when I think about it from there, from a person, I say, oh, well, I wonder whether the mosaicism might have been a factor in why he's really doing and functioning quite a bit higher than we see in Fragile X. So there's no crystal ball that will tell you what's coming, but kind of retrospectively, you might be able to say, yeah, I could see maybe it's because of mosaicism. But you can never be quite sure, because I certainly have met individuals without mosaicism who function quite high as well.
Okay. Well, how about we'll take one more. This is one that that you kind of addressed, but I'm curious too. My son's blood report stated he has an approximate repeat number of 250 and is 50% methylated. Are there further tests that can be done to get details such as where in the body the methylated protein is being produced?
That's a great question, and I do get that actually quite a bit. So to answer the question very directly, there is no test to find out where in the body the protein's being expressed. It's not like there's a scan of some sort that would highlight the areas right now on a clinical basis where the protein's being expressed. There are studies to try to measure the amount of protein or detect if protein is being expressed. Those are not, those are being done pretty much on a research basis, so there's not like a clinical test that you can order to check for protein. So the answer is you really can't tell, and really knowing that lab result and knowing that there's a high percentage of unmethylated FMR1 genes there, you make the assumption that there's protein being expressed. Unfortunately, we know it's being expressed probably in the blood. We just don't know very much more about where it's being expressed, and it must be very frustrating not to know, but on the other hand, your child is going to be your guide, and you're going to see what are the things that he's really doing well at, what are the things he's struggling at. And so it really comes down to the same types of recommendations that we would have for any child with Fragile X, regardless of mosaicism.
Okay. Can I ask you one more quick question?
Sure. I'm glad there's a lot of interest.
There is. So this isn't really about mosaicism, but I do see this question on Facebook frequently. We've been able to track our family pre-mutation back to my mother, but my grandparents both have normal X chromosomes. Is it possible for Fragile X to randomly start in a family? My mom has a pre-mutation, I have a pre-mutation, and my son has a full mutation.
So Fragile X, as far as we know, does not randomly start out of the blue in a family. It happens in a family through a series of successive expansions. So when the person writes that her grandparents have normal chromosomes, what I would wonder is, does one of them actually have an intermediate, one of those like expanded types of expansions of FMR1 that's bigger than expected, so 45 to 54 in that range. So it wouldn't have been reported out as a pre-mutation, and certainly not a full mutation, but it was bigger than we expect. And then when that grandparent went on and had children, had the daughter, it expanded and it squeaked over into that premutation range. And then in the next generation, again, it went to the premutation range and then ultimately to a full mutation. So these things don't happen out of the blue. Fragile X has got an unusual pattern that way too. There's nothing in Fragile X that happens out of the blue. However, we think that many generations back, some of these premutations are the descendants of intermediates. So they weren't quite normal size. They were expanded beyond what we would expect to see, and that's why we have that special category for them, these intermediates. But then in the next generation, they grew again and became premutations. So it's not like the premutation came out of the blue. It came from an intermediate. So I would wonder whether that would be the situation, and it could be, depending on how far long ago the testing was done, that the grandparents' tests just were reported as normal because it didn't show a premutation or a full mutation, and they didn't report out an intermediate.
Gotcha. Right. There's a couple more questions. Please email those to me, and if you can, include your test results, and then we'll either guide you to a local genetic counselor or we'll look at them. Speaking of, Brenda, what's the best way for families to find a genetic counselor close to them who is knowledgeable about Fragile X?
Oh, great. I'm glad you asked that, Jane. The easy answer is, and I'm really pleased to see the new website from the National Society of Genetic Counselors. They have a patient-friendly website called aboutgeneticcounselors.com. And if you go there, it walks you through genetic testing, it walks you through a number of different kind of informative areas, but there's a section there on finding a genetic counselor in your area, and you can put in your zip code. So that's number one. You can go to aboutgeneticcounselors.com and try to locate somebody in your area. If it depends on what you're looking for. If you are thinking about planning a family or are pregnant, you're going to want to look for a prenatal genetic counselor. If you have a child with Fragile X and you're looking for information to help your child, that would be a pediatric genetic counselor. And so you want to look in those areas. If you are an adult and you're thinking about carrier testing, I would reach out probably to a pediatric genetic counselor who might be able to guide you to an adult geneticist, or maybe they also see adults in families with Fragile X. So that's number one. You can find genetic counselors in most areas. And that's a good way to find certified and licensed genetic counselors who are trained to provide this information. One important thing to know, though, there are thousands and thousands of genetic disorders, and I often say this to people, and so sometimes people feel like, that genetic counselor really didn't know all the details about Fragile X that Brenda Finucane does, or some other genetic counselor who spends all day working on Fragile X. Well, I have to say, in defense of my genetic counselor colleagues, they provide information that is generally accurate and are trained in this area, but it may not be their area of expertise. If somebody asked me to provide genetic counseling for cancer, I would be up a creek. I could provide a little bit of information, but not the in-depth kind of information somebody might be looking for. There are not large numbers of genetic counselors with expertise in Fragile X. However, most genetic counselors, certainly working in prenatal and pediatric, would certainly know about how to get testing done, would know about the various types of tests, would know the basic inheritance of Fragile X. So it really, what I would say is, what are your questions and what are your expectations? And if they have to do with things like development and really understanding behavior in Fragile X, probably a genetic counselor is not your first stop. You should be seeing a developmental pediatrician or a psychologist familiar with Fragile X. So just make sure you understand what you want to get answered by a genetic counselor who can help you with the genetics and the testing and explaining mosaicism like I just did. But again, keeping in mind that, Fragile X is one of thousands of disorders, and the person may not have extensive firsthand experience with Fragile X.
Okay, great. With that, there's just a couple, any other questions, feel free to email them to me. It's Jane JAYNE@fragileix.org, and then I can confer with Brenda on the answer. So again, Brenda, thank you so much for sharing your expertise with us. It was, you know, I was wondering if we could spend a whole hour on mosaicism. Obviously, we can. So.
Obviously, and that's just mosaicism. There's so many different aspects to genetics.
I know, and so I know that, you know, to the families who are on, if you have other, you know, further questions that we can use in other additional webinars, please send them to me. And I do want you to know that Brenda will be at the international conference this summer in Cincinnati, so I hope you'll be able to join us there. And she is going to do a session on genetics at the conference too. Before we sign off, I want to let everyone know these webinars are made possible because of your contributions to the National Fragile X Foundation. We are working to schedule additional webinars, so please watch your emails for upcoming information. And again, the registration for the International Conference opens in March, so watch your emails and check the website. And, you know, and I said, you know, if you ever have any questions, shoot me an email. And Brenda, thank you. I mean, thank you doesn't even begin to tell you how much I appreciate all the information that you shared with us today.
Oh, sure. Great. Anytime. And again, be in touch with the foundation and we'll try to get your questions answered.
All right. There you have it. Today is Washington's birthday. Hey, everyone. Have a great afternoon, a great evening. We'll talk to you next time. Thank you.
All right. Bye-bye.